that's why you don't find, we do not have much of probiotic curd or something like that regularly coming in market. It is very difficult to maintain. Okay, so these are some of the health promoting effects attributed to probiotics, and what are they? One is alleviation of symptoms of lactose intolerance. Mostly people who are lactose intolerant, they are not able to, digest lactose, mainly because they don't have lactase. Now, this probiotic organism will all be able to convert lactose to lactic acid. So that is a good point here. Serum, it helps in serum reduction, cholesterol reduction. And we'll see in the ensuing slides. Anti-cancerous effect it should have. It should improve constipation, diarrhea, and also stimulate. So we've been seeing how it stimulates immune system in the previous two slides. So it's a continuation of that. So this probiotic also will produce certain enzymes, and these enzymes helps in digestibility. It helps in enhancing nutrition profile, reducing gastric complication, etc. Also, it produces GABA and CLA. Now, what is CLA? Conjugated linoleic acid, which is, it is an antioxidant. It helps in reducing cancerous cells. Purely, it entraps your free radicals. GABA, no, GABA is nothing but gamma aminobutyric acid. This actually naturally it is present in the cytoplasm of the presynaptic section region in our neurons. Between two neurons, you have a presynaptic region. It is present there. Now, what is the importance of GABA? GABA actually has a calming effect on us. Naturally, we have the calming effect. So what happens? It's like an antidepressant or antihypertensive. So when we are, excited, some people know when they are very excited, what happens? The nerve impulses are passed on. So when it is passed on, it's complete. There is always excitability of the neurons. So the brain is always in that hypertensive position. So for that condition, people are prescribed GABA, synthetic GABA, which helps them to calm down, mostly present in many of the plants. By nature, we have this. So if we are very, excited, very excited, then, this nerve impulses are on a spree. They keep on moving. So they have found that yogurt, which contains Streptococcus thermophilus, produces about 8.3 grams per liter of GABA, which is a wonderful thing. So it helps in comforting us. So that's why I feel curd rice, when we have curd rice, it's a kind of a comfort food. It helps you calm down, especially when you're, irritated, angry, or, you like to take. And of course, in our Indian traditional food, you see, after the end of the meal, it's only the curd rice. So it gives you a calming effect. Okay. So that's about GABA. Then you have organic acid. Lots of organic acids are produced, which will help us along with acetic acid. Some of them produces along with lactic acid, acetic acid, like in bifido. So they help in giving lot of other, they help as energy source for the muscles. They help as carbon source for the colonocytes, etc. Then also vitamins. Of course, they produce a lot of vitamins. Bile salt hydrolyzed. We are going to see in the next slide how it does. So that's an enzyme which it should possess, and of course, a lot of bioactive peptides. So I was talking about cholesterol, how it reduces cholesterol, or how it should have bile salt hydrolyzed. So cholesterol is a precursor of bile. Okay, so we have a lot of cholesterol. It's supposed to be a precursor. Before the bile is formed, it stays as cholesterol. Cholesterol gets converted into bile. So what happens when the bile comes? Bile has got taurine. Taurine is an amino acid which is conjugated with bile and glycine. Now, taurine is present, I'm sure you all must be knowing, in meat, red meat, fish, meats, but not much in plant, in vegetarian food. Okay. So what happens? Taurine is produced. They are produced in the cells, and when they are produced by the body, this goes as it's bound to bile, so it gets defecated. So of course, in the feces, when this probiotics have got this particular enzyme, I said bile salt hydrolyzed, what they do, they deconjugate the bile and they remove glycine and they remove taurine. Taurine goes back to the system, which is very needed for it helps our retina. So for our photo receptors, especially in the retina, this amino acid is needed. So taurine is taken back into the system. So when the cholesterol is being, when the bile is being deconjugated, how's the cholesterol reduced? So when the bile is deconjugated, the system, our body sends messages to the liver and to the brain saying that bile has been deconjugated. And of course, why do you need bile? You need bile to emulsify the fat. So you don't have bile in the body, you need bile to emulsify. So what happens? Cholesterol is derived, especially the LD, the low density lipoproteins. They are derived from the system, from your arteries, from your bloodstream. It goes to the liver. From there, it gets converted, goes to the gall, and then to the your duodenum. So this is how cholesterol is drawn. So cholesterol is drawn, reduces your blood pressure, reduces your, arterial plaques, reduces cholesterol, and thereby it keeps on breaking. So that's why every day you should have, we say, live microorganisms which, when taken in adequate number, get you that health benefit. So you'll have to take every day. Not one day I take probiotics, one day I take good organism, and I'm getting my cholesterol lower. No, this is a continuous process. So this is how your cholesterol and your bile salt hydrolyzed work. Now coming to colon cancer, how does it have? Now coming to cancer, and in special thing, because we are talking about GI tract, we are going to talk only about colorectal cancer. So colorectal cancer is one of the most important cancer which has been diagnosed. We are racing third in the world, with the CRC, and mostly it is present because of the oncomicrobe Clostridia, which is present in the colon. So there is a complex network, and what is a complex network is because there is a lot of pro-carcinogenic substances and bacteria. For example, we take nitrosamines, we take a lot of preservatives, take a lot of polyaromatic hydrocarbons like your barbecue, your all kinds of sauces. Those additives, etc., are not getting degraded. They have the taste; are not getting degraded. So when they don't get degraded, they go to the colon and settle there. They form the nidus. They are like pro-carcinogenic substances. They form the nidus to cause mutation and to cause your microbes to grow, your Clostridia, and also cause certain mutation. There is DNA oxidation, and you have every chance for CRC to develop. So this is how it develops. And of course, there are a lot of free radicals, you are also exposed to a lot of pollutants, etc., which can cause. So you have lot of reactive oxidative oxygen species, which we say as ROS. And this ROS, by normal, it is there in mitochondria in your cells. But when there is an exogenous surge of it in terms of pollution, smoking, that's why you say you also become a passive smoker, because you keep inhaling. And those pollutants go straight into not only to your lungs, it gets through your bloodstream, it goes. So all those will cause damage to your colon area. We also have something. So what happens in this basically is, you have something called this RAS, which is called as, it's a protein, because it was discovered in rat sarcoma. We call this protein RAs. So this RAs, what it does is, it's a protein which is bound to guanidine triphosphate. Okay, so this guanidine triphosphate, what it does is, it keeps, multiplying. As it multiplies, the cell grows. The cell division is there. Okay, so one cell becomes second, and then the GTP, that is the guanidine triphosphate, which is attached. The RAS is attached to that. Okay, RAS protein is attached to that. So once one cell is doubled, what happens? The cell division must stop. We call that as apoptosis. Okay, the cell division must stop. But what happens in case of mutation, or in case of when there's lot of ROS, when there's lot of pollutants, the GTP, which is bound to RAS, does not stop. The RAS becomes kind of let's say oncogenic. The protein, or let's say the gene which is producing this RAS, becomes oncogenic. That means it is prone to form cancer. Okay. So what happens? The GTP does not disengage itself to GDP, which is guanidine diphosphate and phosphate. That means the cell is dead now. Okay. So there is another cell division. So it does not happen. So it keeps on multiplying. So imagine if the cell keeps on, it's immature and keeps on multiplying. There's no apoptosis, and that's how cancerous cells are formed. So cancer tumors are formed. Can you see the big lumps? So there is no cell apoptosis, no cell death. There's a lot of immature cells being formed. So this happens when the particular candidate is exposed to some mutation or some DNA problem which happens in the codon 12, 13, and 61 of this RAS oncogene. The gene which produces this RAS protein becomes an oncogene, and this protein which is formed from this oncogene binds to GTP and does not stop apoptosis. Keeps on becoming bigger and bigger till you have a malignant tumor. So this, by having your probiotics, how does probiotic help? So when probiotic help, it prevents all this, breaks down all this nitrosamines, breaks down all this procarcinogenic substance. It actually prevents this metabolic pathway where this oncogene is formed. So by that you have, so there's no free radicals. So by that you have GTP forming GDP. So there is not much. So it is not like 100% you take probiotics. Studies have shown that the more you take probiotics, the more risk for RAS to become gene to become an oncogene is less. So this is how it helps in preventing anti-cancerous. Okay, and then of course you have lot of intestinal enzymes like beta glucosidase, which you have in the colon, nitrate reductase, azoreductase, glycones, creosotes. All these are there in the. You have the production of all these enzymes by a lot of onco microbes or intestinal microbes in the large intestine, and this causes the nidus for colon cancer. And this probiotics, mostly some of the probiotics we can say that they are present in the small intestine. Some like bifidobacteria, they find more habitat in the large intestine. So this helps in preventing. So they prevent and disengage these organs, they degrade these things, and they are all sent out through the system. So probiotics in CRC, they have found they were depleted. So when their studies had found out, they found people who had colon cancer had very less of probiotics in the colon. They almost nil probiotics. So they came to a conclusion that if there is colonization of these organisms, it would help increase more and more of mucin. It will increase more and more of tight junctions. So we will see what those tight junctions are. So there are no leaky guts. So your epithelium are all tight. So there's no leakage of toxemia in case if there is any infection from the intestine into the bloodstream, all that. But there's one, two, I won't say completely, there are one, two, concerns which we are raising is, one is bacterial translocation. When you eat it, because they are all so minute, they should not be able to, if there is any leaky gut or something, the organism should not migrate to some other part of the system. And especially in endocarditis, especially in people who have, which we call as small intestinal bowel obstruction, SIBO we call. So people who have very small intestine, they have, because of this, they have fogging of brain. So in those conditions, we cannot, be 100%, we cannot be sure of prescribing them this. So they cannot take this. So these considerations, when they have endocarditis and when they have SIBO and when they are very prone, to conditions like, toxemia, then we should be careful in prescribing these organs. So this is what I was saying. There are a lot of leaky ducts, and leaky ducts can be due to the reduction in certain type proteins like claudine or occludine, which will cause there will be a gap between the villi, two sets of villi. So when there is a gap, what happens? You have all these fragments or all these toxin system getting inside your systemic system. So this is how I was talking about. So this is when your gut is not healthy. So what happens? You have gaps like this. You have gaps like this. So any one of the organisms, if they produce any toxic substance, they can go straight through this into your bloodstream. Whereas if you have probiotics, they will produce this occludine or claudine. They are protein. So they produce certain proteinaceous compounds which will help in tightening this junction. So when you are tightening this junction, you do not have this kind of infection being sent to your system. Okay. So this, I was talking about this only. This cancer will be initiated if there is high amounts of, intestinal enzymes or glycones or azoreductase, all these enzymes which are present. Also, if there is some organism will produce hydrogen peroxide. Now, hydrogen peroxide, you all know, it can oxidize, or it has oxidative damage to the DNA. So if it damages your DNA, naturally there will be mutation. So some organisms, when they produce catalase in your system, especially in the large intestine, they are helpful in disintegrating your hydrogen peroxide. So these are called as biomarkers. Okay, so they can be used as biomarkers to alleviate colon cancer. Okay, especially your RAS-P21 oncoprotein. So this is what I was telling. So the more and more of probiotics, if they have supraoxide dismutase or catalase, all these will help in the reduction of free radicals. They help in like your glutathione reductase. If they have all this, they will prevent carcinogenic substances. Okay, so these kind of hydrogen peroxide, etc. All these are present mostly now because we are exposed to pollutants, lot of different kinds of climatic changes, where a lot of pollutions are there. Earlier days, we didn't have so much of pollutants. The exhaust of motor vehicles and smoking and fumes, etc. All these will definitely will form free radicals in our body. So when these free radicals build up, then we have every chances of thyroid cancer, chances of pulmonary cancer, pancreatic cancer, etc. But by organisms, especially this is for GI tract. GI tracts, I'm talking mainly about colorectal cancer. So in case you take in food which has got a lot of additives, etc., and if you have probiotics, they help in reducing the onset of any one of the diseases. And of course, mycotoxin is one carcinogenic substance which is not only carcinogenic but also tetragenic, and also it's believed to be oncogenic. So tetragenic because the next genotoxic also because the next generation, especially your sex chromosomes will be affected. The next generation is affected. So mycotoxin can easily be there are many theories wherein they say organisms, probiotic dairy organisms, especially like propionibacterium, which you find in Swiss cheese, they help in binding. What they do is they bind to the toxin, mycotoxin. So by binding to the toxin, they make the toxin unavailable for being it free. So only if the mycotoxin is available in free form, it can damage the DNA. If it's not available in free form, it can. So this is one of the important things of probiotic food. There's one more important thing called quorum sensing or quorum sensing. I'm sure you would have heard that. So quorum sensing is what when a large number of collective population of organisms, pathogenic organisms are present in the body, they produce virulent factor. Okay, like addition. That's how you get. So you have one salmonella, you're not going to get typhoid. But you have a many number of salmonella, then they produce the toxin. You have many number of salmonella vetiverdi, then they produce food poison. So this is the collective number. If the population is more, so quorum sensing is one kind of a, let's say, receptor or a response this probiotic has. So when they know that the population is more, they send. I have probably in the So they send certain. They send their antimicrobial properties. They send their peptides to kind of it's like a crosstalk. So they listen to the crosstalk and they prevent. So they kind of form addition. They break that kind of a collective population. So there is no formation of biofilm. There's no formation of lethal factor produced. So this is how quorum sensing is employed by the probiotics. So this is what I was telling. If there's homeostasis, you have big villi, you have dendritic cells, you have galt in very good condition, you have all the active, macrophages especially present. But when you have a dysbiosis, you can see the tight junctions are all broken. Organisms are trying to come in. Then if there is more and more, especially in irritable bowel syndrome, you have chronic inflammation. The macrophages are a bit confused also because they're always fighting. So there is not enough organisms or enough memory cells present. You have a lot of organisms, so they're all broken. The mucus lining is broken. Probiotics are not there, so they are not producing the adequate mucin too. So you have this chronic. So you can see the villi shortened and the organ, the particular candidate, whoever intestine is this is going in for disease condition. So these probiotics, what they do, they ferment. They also need some food. So how do they grow? So there are certain products called as prebiotics, okay, which we will see in the subsequent slide too. Prebiotics, and these prebiotics are foods which are not digested by our body, our enzymes. They are present as such. But however, they are worked upon by probiotics, and some of the common salts, and they produce short chain fatty acids like your butyric acid, acetic acid, propionic acid, and these serve as food for the colonocytes, for the colon. So it keeps that forgotten organ active. It keeps it more healthy. It produces more about vitamins. It helps in modulating some of the peptides. It helps in vitamin synthesis. It basically shapes the intestine. So this is a picture of how the villi is given. It's a cross section. It's a LS view. So you can find the probiotics when they are present. They present. So there are a lot of IgA immune response is there. IgG is response. IgG is also present, which is one of the biggest. So they help in the memory cells. So they're always keeping the memory cells and also the toll-like receptors active. So because they're active, and they also produce, they are also involved in quorum sensing. You have a lot of peptides and receptors. Can you see the toll-like receptors all present? So they help in preventing the stabilization of any of the chronic inflammation. So they're always on check, and the system is healthy. So this is what I was saying. So this they help in IgG, B cells, production of IgA, and therefore, even if there are any pathogens, they get caught in the mucosal layer, which acts as a barrier. So there are certain adjuvant effects. Adjuvant effects means there are certain additional effects, which is not the main effects, but however some effects. What are the additional effects or the adjuvant effects of probiotics? So they improve antigen presentation. That is, even a bit of bacteria or fungi which is left, okay, during digestion, if the bacteria would have killed. But even if a bit is there, these help in presenting this. They present that to the T cells. By presenting, they keep the memory active. So the immune system is active. They recognize this particular bit of bacteria, and they are active and they are ready to fight. So they keep the host defense mechanism, especially the guard, very active. Then they also help in the transport of antigenic material. So if there is any bacteria, is present, they have the ability through their mucus or through their sensing, through their peptides, they sound the gut mucosa. They also help in stimulating the immune cells, especially the B cells. And also, whenever oral therapy is given, oral vaccines or oral therapy is given, we always prescribe certain probiotics. So this is one important small component which I will talk about is gut-brain axis. So probiotic, now they say something like GAPS, which is gut-associated psychological syndrome. So this word has been coming often as GAPS or gut-brain axis theory. So what they say is when your gut is healthy, your brain is also healthy. That's what. So people who are like mentally depressed or mentally not very healthy, autistic, you will find them to always have either constipation or diarrhea. That's because the gut system or gut health is not happy, especially the beneficial organisms are lacking. So how they are lacking is, there was one study where they said is, serotonin, I'm sure you've all heard, serotonin is present, which ignites your happy centers. This is the particular hormone which helps you keep very active, it helps your brain active. So tryptophan, which is normally, it's just an amino acid, we all need the amino acid, but sadly it is not much absorbed by the system. It is found in the colon. It is defecated on. But plants like aloe vera, that's why aloe vera, they say, is very good. Aloe vera and probiotics, they help in absorbing this tryptophan back. And tryptophan is a precursor for serotonin. So when more and more tryptophan is absorbed, more and more serotonin is found in your system, which helps your which helps your brain, which helps your brain. So they have found a study where they found people who have Alzheimer's disease, they have more amount of tryptophan, I think phenylalanine and tyrosine in their blood, and also in their feces, lot of calprotectin, which is being wasted. Okay, these amino acids they found in their feces, which is being wasted. That means they're not absorbed. So they found that the more and more of probiotics are there, more and more absorption of tryptophan and phenylalanine and all this, calprotectin, everything goes back into system. So this is one of the study tips. There's another study involving Parkinson's disease, which I happened to see a few months back, and what they found is these gut microbes, they if these like probiotics, they are known to produce a lot of biotin, riboflavin, etc. So Parkinson's disease happens when there is reduction of biotin and riboflavin. When the particular candidate has got less. So now you see a lot of cases of Parkinson's disease being seen in a lot of patients, and this they attributed because there is a decrease of these two vitamins, and because there's a decrease of these two vitamins, there's a decrease in short chain fatty acid, which keeps the healthy layer in the intestine. And one of the causes for this is because they found the common chemicals which we use for cleaning, like a liquid soaps or detergents, which we clean in which we use for daily wear, no, or kitchen utensils. We don't wash them well, so when they form a biofilm, these toxins, they are like toxins, the biodetergents. No, when you don't wash them, sorry, they're not biodetergents, they're synthetic detergents. When you don't wash them well, they form a kind of a biofilm, and this leads to the overproduction of alpha-synucleic fibrils. They are kind of a fibril-like substance which goes and sits on your dopamine center, which is present in your substantia nigra in the brain. So it goes and sits there. So when it goes and sits there, it's kind of a muffled. Okay, so there is loss of motor activity. There is loss of there's loss of activity and there's debilitating. This kind of solution goes through your bloodstream. So that's why they say wash it well. And I was also left to wonder whether earlier days they used to use only the ash, wood ash or whatever ash to wash. But now, no, that's kind of a considered very gross. Nobody wants to dirty their hands with the ash and wash, So now it has gone, and now people use only, detergents and easy washable and flavorful detergents. Okay, the ads comes out. So might be these have to be washed well, otherwise they will produce this, and more of this helps in Parkinson's because it is going and occluding there. This is, by the way, this has nothing to do with probiotics, but because of this reduction in riboflavin and biotin, more and more amount of cases are coupled with this. This is one of the things I thought I should tell. So prebiotics, which I was telling, is they are food for probiotics. They are producing a lot of short chain fatty acid, which helps these organisms to grow. And you have abundant in banana in the form of inulin, in as arabinose and xylooligosaccharides in ragi. You have in cabbage, you have in onions, you have in garlic, you have in so many other, soy. You have in all this, of course, lactulose, you have in milk. All these are prebiotics which do not get digested in our system, but they are used as food. They help the organisms to grow. So with this, I come to almost the end of my talk. And this is a study which we did in our college, where we found some probiotics which we isolated. We did all the characteristics to study whether they are probiotics, and few of them were probiotics. We saw this. This is how they look, and we use them to after neutralizing, because for probiotics to be called, they should have some antimicrobial substances. So we checked against certain organisms which cause mastitis in animals. So we found them to prevent those organisms. So by that we said we could use them as dips to prevent, as a preventive measure for development of mastitis. Thank you very much. I hope I was able to convey whatever I had. And if You have any questions, you can ask me.
Thank you, ma'am. Participants, if you have any question, you can unmute your mic and ask, ma'am.
Good evening, ma'am.
Good evening.
Ma'am, I want some suggestion, ma'am.
Yeah. Yes. Can you introduce yourself and tell me so I can? Hello.
Hello.
Yes.
Ma'am, அது பத்மஜான்னு நினைக்கிறேன்.
Yeah.
Okay. Padmaja. She is a student, ma'am.
Okay. She can
Padmaja?
ask it.
She is not online.
Okay. All right. She can still mail me and ask me. Anybody else? Would you like to?
Anyone else?
So I read in an article wherein I found see lactobacillus astrophilus, that again is a probiotic. It has got mannose on it.
Okay.
So this mannose, when they did some in vitro trials, they found AIDS virus to bind to that particular mannose. So they could find that this virus, if it's present, astrophilus helps in binding. Okay, so because of the mannose content. So this virus needs mannose, so it goes and adheres itself. So by that, you can use this organism, like kind of. And I also say whenever you take a photograph, you always say cheese. Okay, cheese, because you need to show your entire set of teeth, and that's because they say after you eat, have a bit of cheese, which will help your teeth glisten. That's because this cheese has got a lot of organisms, good organisms, which helps you clean your teeth, in the sense it doesn't give space for the bad organisms, especially Streptococcus mutants, which cause gingivitis. It doesn't allow. So this will go and attach itself. So you don't have any foul breath. That's why they say cheese. I don't know whether it was on a fun part, but I found that very interesting. So that's how the people say cheese and not any other.
I think now Padmaja is in.
Yes,
Hello?
ma'am. Hello, ma'am.
Yes, hello, Padmaja.
Yes, ma'am.
You
Ma'am,
can ask your
I'm
question.
currently in masters, ma'am. So, what would you suggest me to do in postbiotics and prebiotics for my project, ma'am?
Yeah, you can take postbiotics too. You can take the bacteriocins, or after it is neutralized, to see that does not have acid. You can work on postbiotics because that becomes very easy. You don't have the error of translocation. That's good. Prebiotics also, because if you're using postbiotics and prebiotics, you cannot use together. Prebiotics should be used along with probiotics.
Okay, ma'am.
So postbiotics are just the metabolites of probiotics. It does not have any live cells. That is postbiotics. So you cannot use prebiotics and probiotics together. Is a good thing because they will work together. Postbiotics can be used alone. Prebiotics, if you are taking it, is for the system inside. You cannot assess it. You cannot assess how well it works unless you have a live organism present.
Okay, ma'am. Thank you, ma'am.
Okay. Thank you. Anybody else? How many participants are here?
இந்த இதுல இருந்து கொஸ்டின் கேளு.
Ma'am, now around 90 participants are there, ma'am.
Oh, that's a good number.
Yes, ma'am.
Just a second. Hello, ma'am. Good evening.
Good evening.
I myself, Josna. I'm assistant professor from JN University, Bangalore.
Okay.
Thank you, ma'am. It was a very excellent presentation. It covered all the functionality of probiotics. Ma'am, I
Thank
have a
you.
question. Yeah,
Yeah.
thank you. I have a question. Is there any genomic markers which we like to get to know about the functionality of probiotics? Is well-known genomics markers.
For
Your comment.
probiotics,
Markers, ma'am. Markers to know
marks.
the functionality of
See,
probiotics.
they're still working on that. You can't be sure about the markers. Uh,
Okay.
I am not working on that. I'm just working on the traits of it alone. But
Okay.
they're still working, but it's all very nebulous only in the laboratory. They can't pinpoint it is due to this. A couple of more, functional properties are coded with one gene. So they are not very sure about it. But right now we are working only with, of course, you can do 16S and you can find out the entire thing. That's different. You can find out where it is and what. But right now, I'm not aware that they are having any markers for particular functionalities.
Yes, ma'am. Yes. Thank you, ma'am. Thank you so much.
Anyone else?
Good evening, ma'am. This is Katyayani from Manakula Vinayagar Institute of Technology.
Good evening.
I have a question about can fermentation play a role in plant-based and alternative protein development?
Fermentation. Yes, it can, depending on the kind of organism you're going to use. So the metabolites will vary. You can use. There's nothing you can use fermentation. You have kimchi, all that is fermented products, not plant based.
Yes, ma'am. Yes.
You can use. Depending on the metabolites will be varying. It won't be akin to milk and milk products. It'll be different metabolites. You'll have butyric acid. You'll have different. Of course,
Okay.
plant based. There are a lot of plant based fermented products available, which also works wonderfully.
Okay, ma'am. Thank you so much.
Okay. So
Ma'am,
anyone else?
if
Yes.
there is no question, we can conclude.
Okay.
Rita Narayan, ma'am, Dr. Rita Narayan, ma'am, on
Yes,
behalf
yes.
of the department, I extend my thanks to you, ma'am, for accepting our request.
Okay.
And you have given excellent presentation, which is in depth also. And you have covered all the things related to gut health, ma'am. Okay. Thanks a lot, ma'am, from our
Thank you very much for the kind opportunity, sir.
Okay. Nisha, ma'am, you can formally say what
Yes,
of that.
sir. Yes, sir. Yes, sir. Okay.
Okay.
On behalf of the organizing team, I take this opportunity to express our sincere gratitude to Dr. Rita Narayanan for her in-depth and technical rich lecture on power of fermentation, advancing gut health and microbiome focused foods. Her session provided valuable scientific insights on how microorganisms respond under different environmental and processing conditions, and also on how these interaction influence fermentation outcomes and human health. The clarity with which complex micromechanisms and their health impacts were explained made the session both intellectually stimulating and highly informative. We are truly grateful to Dr. Rita Narayanan for dedicating her valuable time and for sharing her expertise. Offer a deeper understanding of microbiome-driven food system that will greatly benefit students and researchers. Once again, we extend our heartfelt thanks to Dr. Rita Narayanan for her insightful and technically impactful session. Thank you so much, ma'am.
Thank you very much, Dr. Nishan. Thank you very much, sir. Thanks for having me on your program. Thank you very much. Thank you,
Thank you.
participants, too.
Thank you, ma'am. With this, I officially conclude the session. Thank you. Thank you, ma'am.